All right, so we'll go ahead and get started. It is my pleasure to introduce Dr. Magdalena Mrokowska here today as our invited speaker for the seminar series for the month of March. So I've known Magdalena for a few years now, where we met at a conference, and we've been sort of exchanging emails and ideas because we had some similar work related to how particles orient in oceanic environments. So just a brief overview of Magdalena, and I'll let her talk about her work soon. So she's currently an associate professor at the Polish Academy of Sciences in the Institute of Geophysics. And prior to that, she earned a PhD in 2014, as well as a master's in science at the Interfaculty Study of Environmental Protection at Warsaw University of Life Sciences and Wageningen University in the Netherlands. And her work primarily focuses on the fate of particles in both marine as well as riverine environments. And she's primarily an experimentalist, and she works a lot with developing tools such as imaging approaches for lab-based observations of particle behavior. And so without further ado, I will let Magdalena begin her talk.
Yeah, thank you, Aditya, for introducing me. Yeah, it's a great pleasure for me to be here, to have opportunity to give a talk. I would like to thank the organizers for inviting me. In this presentation, I'm going to show some observations from quite extensive topic that I took up with my collaborators some time ago. Let me introduce the team, the project team. I work with Professor Anna Cztoń-Maziopa from Warsaw University of Technology and a group of our students, Karolina Dzień, Tomasz Habło, Monika Pękalska. We also have a group of project collaborators, Dr. Arkadiusz Antonowicz, Professor Maciej Dąbowski, Professor Sylwia Różańska, Dr. Ewelina Wambiel-Wytykowska, and Peter Fischer, and those people contributed to some specific parts of our project. The motivation of our investigation is the significance of the dynamics of particles in marine environment. Particles in the ocean, such as minerals, marine snow, and microplastics, serve as vectors for carbon, nutrients, and pollutants. And it's essential to understand the processes that influence their fluxes. Some basic questions are: how fast do particles settle? How long do they reside in the water column? And how do they interact with one another? Several factors influence dynamics of particles, including surface waves, turbulence, and the stratification of the water column. When it comes to stratification, it causes some interesting sinking behavior, as an isometric particle sinking through density gradient reorients, and its velocity varies due to buoyancy-induced effects. Here I show how it looked like in some of my previous experiments with disk into layer systems. Particles sinking in steady motion in upper homogeneous layer decelerates at density transition, not only due to fluid density increase, but also due to stratification-induced drag resulting from added mass effect from entrained fluid. Reorientation is caused by the deflection of isopignals and pressure distribution around the particle. Deceleration and orientation instability are followed by wake evolution, which is revealed here by dye visualization. The disk is in a stable horizontal position with almost no movement until the major portion of the wake separates from the disk and upward jet forms. With a kind of a swelling due to internal wave formation. And these results were quite unexpected, and then further questions arise. And as these conditions in water column could be even more complex in natural water, we addressed what happened if exopolymeric substances appear, which are frequently reported in stratified systems. Extracellular or extracellular polymeric substances, or EPSs, are secreted outside cells. They are slimy gel-like substances. They are either tightly bound, either tightly bind or loosely associate with the producing cell. Exopolymers play a crucial role in supporting colony structure and providing protection against environmental and biological threats. They can detach or dilute into the surrounding water, and microalgae and bacteria are the primary but not the only producers of exopolymers. Other organisms, such as jellyfish, corals, macroalgae, also contribute to exopolymer pool. However, microalgae are dominant producers due to their abundance and ability to form blooms. And today, I will present a different perspective on the role of exopolymers in the ocean, focusing on how they modify seawater properties and influence the sedimentation of solid particles, rather than examining how carbon fixed through photosynthesis cycles within the environment. Exopolymers consist of polysaccharides, proteins, lipids, nucleic acids, with polysaccharides, and polysaccharides are the most abundant fraction. These long-chain macromolecules are often depicted schematically in a spaghetti-like structure. Some of them are linear, others are branched, containing shorter side functional groups such as carboxyl or sulfate groups. In seawater, exopolymers tend to aggregate. They cross-link to form a physical gel, a three-dimensional matrix of weakly bound macromolecules. These bonds are formed by few mechanisms. The first is electrostatic interactions. Seawater is a high ionic strength environment formed by several salts, and ions present in water interact with exopolymers, especially with polyanionic structure of polysaccharides. And additional crosslinks are formed through hydrogen bonds, van der Waals forces, and hydrophobic interactions. And this way, so-called marine gels are formed. In the water column, marine gels span a wide range of sizes across several orders of magnitude. They are truly dissolved exopolymers that aggregate into nanogels, microgels, and macrogels, forming organic matter continuum concept. Exopolymers have a unique ability to form gelatinous associations of various forms. They densely cross-link in a biofilm, which is exopolymeric matrix embedding microorganisms. Further, when exopolymer macromolecules are dissolved in seawater, they spontaneously aggregate into self-assembled microgels, and these forms exist in dynamic equilibrium. In seawater, exopolymers exist in various dilution regimes, ranging from truly diluted states, where macromolecules, the coils exist without touching each other, and then they are from denser and denser structures to entangled states. These elevated concentrations of exopolymers are particularly intriguing, both in a small scale, in the form of biofilm, and on the scale of the water column. This happens during mucilage events when exopolymers form dense volumes of mucus across water column, including sea surface foams and bottom mats. Exopolymers affect various processes. From a microbial perspective, gel-like exopolymeric networks influence molecular diffusion, nutrient gradients, motion of microorganisms. And meanwhile, exopolymers dispersed in the water column impact small to large scale processes such as turbulence, surface wave formation, and sedimentation. And here, let me highlight a recent fascinating discovery on the role of mucus tails formed around sinking aggregates from Professor Prakash lab. While this presentation focuses on different situations when particles settle in mucus-rich seawater. Effects of exopolymers are related to the rheology of exopolymer-rich medium, which characterizes the mechanical response of the medium to applied forces. Rheology is the branch of physics studying the deformation and flow behavior of materials. And two rheological properties are essential in exopolymer-rich systems. They are flow properties and viscoelasticity. And let me briefly characterize these properties. Viscosity is a basic material property of liquids. Dynamic viscosity reflects flow resistance due to frictional forces between fluid molecules that appears upon deformation by shear stress. In a simple Newtonian shear flow, the shear stress is linearly proportional to the rate of deformation of the liquid, that is the shear rate. Seawater is a Newtonian liquid, and the Newtonian viscosity is constant for a given temperature, salinity, and pressure. When exopolymers are introduced into seawater, they form a structure that evolves with increasing deformation. At low strain, macromolecules remain in a coiled form, showing Newtonian behavior. As deformation increases, these macromolecules progressively stretch and disentangle, causing a decrease in internal viscosity. Eventually, the macromolecules reach a point when they no longer undergo significant changes and the flow returns to Newtonian behavior. This shear-dependent viscosity is referred to as apparent viscosity. Viscosity characteristics depend on specific structural properties of exopolymers and on their concentration. And to briefly sum up this slide, viscosity in exopolymer-rich water is higher than that of typical seawater, and it is not a constant value, so it's quite different. The other property of exopolymer-rich seawater is viscoelasticity. Viscoelasticity is a property of material that exhibits a combination of liquid-like and solid-like behaviors described by the deformation of material subjected to the force and recovery after force removal. Viscoelasticity refers to the ability of material to show both viscous and elastic characteristics. At one extreme, liquids are characterized by viscosity, while solids are characterized by viscosity, and solids by elasticity. Exopolymers, however, form intermediate states in seawater, and our focus is on viscoelastic liquids. The nature of viscoelasticity is captured in the creep test that demonstrates how viscoelastic material deforms under force. When the force is removed at time instant t1, only partial deformation is recovered instantaneously, and the recovery is continuing over time. Now returning to the effects of exopolymers on various processes, here are some examples that illustrate significant role of exopolymer rheology over scales. The first study, led by Oscar Guadayol, highlighted heterogeneous viscosity around plankton cells, which affects the diffusivity of nutrients and bacteria. The second example from Professor Stopper lab showed that the viscoelasticity of the exopolymeric network influences bacterial colonies before biofilm is formed, with some surprising coordinated behavior observed at low exopolymer concentration. And the third example shifts to macro scale. It shows locations where the bulk rheological properties of seawater were measured, along with sample locations of mucilage events where the non-Newtonian properties of seawater are likely. And these studies highlight the concept of biologically modified rheology of seawater introduced by Professor Ian Jenkinson and further verified in the field by him and other researchers. And increased viscosity is the primary indicator of biological modulation of seawater medium. This concept divides seawater into two components: one, physically controlled by temperature, pressure, and salinity, and another biologically controlled by organic matter, such as exopolymers. The latter refers to viscosity that exceeds typical seawater levels, and the ratio of biologically controlled to physically controlled components is termed excess viscosity. Now let me briefly move to particle sinking in seawater to see how exopolymers can change its dynamics. Sinking of a particle in a classical Newtonian framework depends on its dimension. Here, sphere diameter, density of fluid, and fluid viscosity. A particle reaches its terminal velocity when the drag force is balanced by gravitational and buoyancy forces, and the ratio of inertia to viscosity governs the sinking process, which is illustrated by the relationship between drag and Reynolds number. Particles with relatively small dimensions or low velocities are dominated by viscosity, resulting in high drag. Examples include bacteria, clay minerals, nanoplastics. As particle size and velocity increases, inertia begins to play a role, altering the flow patterns around the particles, and drag is reduced. And this is a Newtonian case. And how about sinking dynamics in the presence of exopolymers? When exopolymers appear, particles experience increased viscosity, which is additionally shear dependent, and elasticity also appears. These biologically modified conditions affect dynamics of particles, which we have studied in lab experiments. In the laboratory, we simulated exopolymer-rich conditions using commercial biopolymers, mainly xanthan gum and carrageenans. They were dissolved in artificial seawater solutions to create exopolymer dispersions. Bulk rheological properties were measured using a rheometer in both shear and oscillatory modes, and experiments were carried out in settling columns which were about half meter high. We used synthetic solid particles with dimensions from 1 to 5 millimeters that are classified as microplastics, but they could be used as convenient models for other types of environmental solid particles. In one configuration, sinking particles were visualized using backlight, and particle motion was tracked to assess instantaneous velocity and orientation dynamics. We also applied particle image velocimetry technique to analyze velocity fields around sinking particles. Test liquids were seeded with fluorescent particles and illuminated with a laser sheet, and images captured by the camera were processed to compute velocity vector fields. And here, let me briefly introduce the main test polysaccharides. The first one, xanthangum, is of bacterial origin. It has branch structure with hydrogen bonds playing a stabilizing role in the molecule. Xanthangum forms a gel structure mainly due to hydrogen bonding between hydroxyl groups. The other one is of algal origin. Kappa carrageenan is a linear long-chain negatively charged polysaccharide extracted from red seaweed. It comprises sulfate groups that gives it ability to form rigid gels when electrolytes are present, while hydrogen bonds stabilize formed network. Gelation includes transition from coil to helix and further association of double helices, which are facilitated by positively charged ions like potassium or calcium. And here are our sample laboratory results. In one of our studies, we compared the dynamics of microplastics sinking in seawater and in seawater enriched with exopolymers. In this study, we tested 14 types of particles, varying in shape and density. They were spheres, rods, disks, blades, and irregular particles. And we used 6 types of exopolymer-rich solutions composed of xanthangum carrageenan and their mixtures. One of our aims was to explore the concept of excess viscosity as a basic approach to quantifying the effects of exopolymers. We demonstrated that the sinking velocity of particles in exopolymer-rich seawater compared to that in pure seawater decreased in a rational manner with excess viscosity. Based on this, we suggest that excess viscosity, a characteristic of particle settling, can be treated as a representative parameter for the first assessment of effects of exopolymers, which are caused by mechanical strength of the polymer network formed in seawater. It's relatively easy to be measured before other rheological characteristics could be measured. Examining the range of Reynolds number shown in this left-hand side figure presents that that exopolymer significantly shifts dynamics of particles towards low Reynolds number regimes. Reynolds number for our test particles dropped from around 1 to 400 in seawater to much below 1 with biologically modified viscosity exceeding seawater viscosity up to 50 times. This suggests that a different approach to hydrodynamics in the hydrodynamics of particles in exopolymeric rich environment is needed than what is applied in a typical seawater in terms of assessing sinking rates and related drags on particles. Another effect is related to the viscoelasticity of our exopolymeric dispersions. When polymer solution is sheared, normal stresses arise in addition to shear stresses. These normal stresses around sinking particle are strongest when where the streamlines are more crowded. They generate torque that tend to rotate an isometric particle into a vertical position. In inertial regimes, inertia, which tend to keep particle in a horizontal position, competes with this viscoelastic effect. And the particle's final orientation depends on whether viscoelastic or inertial effects dominate. And in videos, we can see that while in a seawater, rod-like particle is sinking horizontally, it tends to vertical position with increasing exopolymer content, which indicates growing viscoelastic effect. And as you can see in the right-hand side figure, we observed various orientations depending on the exopolymer concentration and also depending on the type of the polymer. It depends on the individual rheological properties of our test polymers. The orientations are essential in drag assessment, which is in fact orientation dependent. I'm not showing it here, but we elaborated on this more in our study in the paper. Now let me turn to another study. As I have already shown, exopolymers tend to form marine gels, aggregates of various sizes. Here we aim to analyze sinking dynamics of spheres and disks in solutions varying in sizes of dispersed gels. We studied two types of solutions, colloidal dispersion and dispersion with predominant content of particulate gels. And here we use xanthan gum dispersed in sodium chloride. Here I present sample data for solutions containing 0.7 mol per liter of sodium chloride, and the content of xanthan gum was 1 gram per liter. Settling velocity and drag exerted on solid spheres and disks were substantially different for the two types of solutions, with the sinking velocity up to 2.5 times higher, resulting in drag coefficient up to 6 times higher for colloidal than for particulate solutions. It means that colloidal exopolymers may retard particles falling in salt water more effectively than particulate gels. Surprisingly for us, bulk properties didn't indicate variations in solution properties, and these flow, actually viscosity curves were alike for both types of solutions. And this property was not useful in interpreting our observations here. However, viscoelasticity properties provided explanation for observations. This was caused by stronger inner structure of colloidal dispersions. It was evidenced in amplitude sweep test, shown in this middle figure. Viscoelastic moduli were higher for colloidal dispersion and showed linear viscoelasticity in a wider range of shear strain. Storage moduli G prime was higher than loss moduli G double prime, evidencing a stronger physical network, while particulate dispersions formed weaker gel with comparable magnitude of viscous and elastic component. Despite inhomogeneities, those dispersions were continuous in a scale of sinking particles, which were of 1 to 5 millimeters. However, in nature, we expect some patchiness of gel-like structures. And indeed, we observed that some dispersions of exopolymers formed structural inhomogeneities with gels of the size comparable to the sinking particles. In such conditions, particles can experience varying mechanical strength, resistance, and normal stresses, which can result in chaotic trajectories, preferential paths, fluctuating orientations, and even trapping of particles due to apparent yield. In the video, you can see a sample trajectory of particles sinking in this type of inhomogeneous medium. Settling becomes unsteady with variations in sinking velocity over time. And such transient behaviors are a current topic of our investigation. Besides sinking velocity, exopolymers may affect the flow field around sinking particles. This is one of the most intriguing non-Newtonian characteristics of the sedimentation process. Studies performed over a few decades have identified flow field patterns that vary from that in Newtonian fluids, and we observed them in seawater with dispersed exopolymers. For example, there is the extended wake, which resembles the Newtonian wake but is wider, and the negative wake, which represents the reverse flow behind a sinking particle. Studies conducted over past few decades have clarified the causes of the negative wake, linking it to a combination of shear thinning, viscoelasticity, and proximity to boundaries. And the key parameters are the extensibility of polymers and the associated elongational viscosity. According to this concept, the wake is shaped by two competing mechanisms: extensional stress with the wake, within the wake, and the relaxation of shear stresses around the sinking particle. And we have confirmed that these effects also occur in seawater contained exopolymers. We also observed the attraction of particles sinking in a doublet that can facilitate aggregation of particles in exopolymer-rich regions. This is caused by weak time-dependent shear thinning of our model solutions, shown in this graph. And flow also developed negative wake here, influenced by extensibility of polymers confirmed in extensional tests, which, okay. So, in our latest study, we addressed settling processes at the periphery of algal bloom. We modeled Newtonian, non-Newtonian transition in two-layered water columns, and our test cases comprised rheological and density gradients. We used seawater of various salinities and xanthan concentrations. And we investigated 6 types of microplastics. They included spheres, rods, and disks. By comparing sinking velocity in exopolymer-rich and non-Newtonian layer and Newtonian seawater layer, we observed that the ratio was inversely proportional to viscosity jump, which spanned over 2 orders of magnitude. At the same time, sinking velocity didn't show any relation with density jump. And this supported the observation that in considered conditions, viscosity effects overcame density ones in controlling sinking dynamics. Further, we related the orientational instabilities to sinking regimes of microplastics in Newtonian and non-Newtonian layers. And the strength of rheological transition quantified by viscosity jump. This figure demonstrates that non-spherical particles were sinking in seawater layer in a transitional regime, in all cases, with Reynolds number between 5 and 100. Meanwhile, in non-Newtonian layer, Reynolds number was decreasing towards creeping motion. And these variations were accompanied with orientational instabilities. We indicated 3 regions representing modes of orientation transformation in the rheological transition. Particles settled in horizontal position in the first region of parameter space, where inertia was significant in both Newtonian and non-Newtonian layers. And viscosity jump here was below 4. In the second region, orientation transformed from horizontal in Newtonian seawater to tilted or vertical in non-Newtonian layer. And here viscosity jump was between 7 and 10. And in the third region, transformation was from horizontal to vertical, and it was identified for a viscosity jump larger than 32. And this parameter space is a tentative suggestion of approaching the characterization of sinking dynamics of particles at rheological transitions in exopolymer rich seawater. And let me summarize these findings, this presentation. Excessive presence of exopolymers induces some non-Newtonian properties of seawater and non-intuitive sinking dynamics of particles compared to typical seawater. So particles reside for a longer time in a water column, which may affect biochemical processes, aggregation, and ecological interactions. The heterogeneous structure of exopolymer-rich seawater induces some chaotic motions that may affect aggregate formation mechanisms. Moreover, exopolymer-rich regions modify fluxes of marine particles, still to an unspecified extent, since oceanological studies on sedimentation are focused on Newtonian conditions. This topic is quite interdisciplinary, so research combining physics, chemistry, microbiology, and plankton ecology is essential to understand and estimate the fate of marine particles in regions which elevated amounts of exopolymers. And what I show today is only the beginning of the research necessary to understand the possible effects of exopolymers on sedimentation processes and interactions of particles, and further developments are welcome. And here goes once again a team that was working on the topic. And here are the references to papers I cited in this presentation. So thank you very much, and I'll be happy to answer your questions.
Thanks, Magdalena. Great talk. And so we can open up the floor for questions. Anybody, you can unmute yourself and go ahead and ask a question if you have. And I will also check in the, you can also put questions in the chat if you want. So while we wait to hear questions, I just had one, Magdalena, can you potentially comment on, like, I would assume, I mean, I know that we've sort of discussed a little bit of trying to make these observations in the field, right? And this is sort of a challenging problem. So from your perspective, can you comment a little bit about this in terms of what kind of measurements you think would be needed, like integrated measurements in the field to make some observations like this?
Yeah, indeed, field, yeah, I can hear myself, so somewhere is
Oh,
field.
maybe.
Yeah, field measurements are very challenging in this topic. I'm not involved in any type of field measurement, so I'm purely a laboratory person, but I can comment this based on the literature and discussions with scientists who are measuring in the field. Yes, in this topic is quite challenging. Yeah, because these samples, when we are talking about these elevated amounts of exopolymers, it occurs during algal blooms. Sometimes algal blooms are quite predictable, but sometimes not. So all the whole logistics of field measurements is sometimes a challenge. Then collecting the samples, and then scientists who are able to or who have just the skills to and equipment to measure the parameters that we would like to know to measure these non-Newtonian properties. So what has been done so far, it was just a measurement of viscosity, which is quite straightforward. And then some oscillatory tests in the lab from these samples. This type of measurements could be done also on a ship if there is a lab, but there are quite sensitive equipment, so this could be a bit tricky. However, generally the methodology for broad analysis of non-Newtonian properties of seawater doesn't exist at the moment. There are some papers, mainly from Professor Ian Jenkinson, who did this. But to make this topic more widespread in a scientific community, we really need dedicated oceanologists, chemists to develop this.
Thank you. Any questions from the audience? So there is a question by Oscar in the chat, and I'll read it out. So Oscar says, excellent talk. Thank you very much. I was wondering if you could discuss what the significance of particle size in your results would be, and if you would expect the same sort of responses for particles comparable in size to phytoplankton cells or marine snow.
Okay, yeah, thank you very much for the question. Thanks. Similar, yes. So the size, yeah, here the size and density is important. As I presented, my particles were in let's say moderate Reynolds numbers regimes. For phytoplankton, there will be for sure in even in smaller. For marine snow, it depends. It's very porous and also could be lightweight. But For plant, it all depends on the content of polymers and on the type of polymers and what type of polymeric network develops in seawater. But for concentrations, as I showed here, which I refer to mucilage events, so very abundant exopolymers in water. Small particles like phytoplankton could be just trapped. Very light particles or very small particles could be trapped due to this mechanical strength of polymeric network. When it comes to marine snow, it depends. It depends, as I said, mostly on the shear stresses that these particles impose sinking in this medium with polymers. Some polymers develops yield, so this is some threshold when threshold value of shear stresses where particles will be trapped. So it is it much depends on the parameters on the medium and particles, but for a smaller, it's higher chance of just for trapping.
Thank you. Any other questions that we have? I just also wanted to let you know that the talk is recorded, and it'll be available on YouTube in the mob channel link. All right. If there are no other questions, I'd like to thank Magdalena once again for her time. I just wanted to make an announcement. Next month, it's going to be Dr. Husho Jiang from Woods Hole, who's going to be giving the seminar. So I hope to see many of you there as well. Thank you, everyone, and thanks, Magdalena.
Thank you.